Okay, so good afternoon, everyone. My name is Liu Xiao-Bin, and I comes from the Fungal Genomics and Evolutionary Lab, so from Biological Research Center. So today I am going to talk about the uncover the secret behind mushroom development. We are on the way. I guess maybe probably most of you is very tired after one day's hard work, and you still have energy to sit in front of your laptop to listen this probably very boring topic. I am pretty sure you are a mushroom fan, so I am glad about this. So let's start my presentation. So firstly, I would like to start from the cultural significance. So probably you realize mushroom actually have a very important cultural significance in Western and in Eastern world. So in Western world, you must know the Mycenae city is a very famous city in Greece. So its name in Greek mythology is probably because the Perseus tried to find a spring in the city, and after he find a mushroom, and then he find a spring beside this mushroom, and so he named this city as Mycenae. So it's related to mushroom in Greek language. And another story is about the mushroom circle formation. And people try to explain the mushroom circle formation because of the fairy dancing in the forest. So after the fairy dancing, and then mushroom circle occurred. So which also called by people as a fairy ring. So this is a very beautiful story. I like it a lot. And in the Eastern world, so in China, the Ganoderma lucidum have a very important symbiosis in Chinese culture. So look at this colorful cloud. So people suppose this kind of colorful cloud comes from the heaven, and people call it auspicious clouds because people think this kind of cloud can bring people's good luck and happiness. So look at the Ganoderma lucidum's fruiting body shape. You can find its shape just like the auspicious clouds. So for thousands of years ago till now, people call Ganoderma lucidum as auspicious plant. So which means the plant from the heaven and which can bring people happiness, lucky, and long life. So you can see from the right-hand side painting. So you can he is a one of our empire about 300 years ago, and you can see he hold a Ganoderma lucidum. So as an empire is not surprised, probably he want a long life. So besides the cultural significance, mushroom production also form a large industry currently all over the world. So based on the FAO statistic, more than 40 million tons of mushroom were produced every year. So, and you can see from China and the Netherlands, the growth of the Flammulina filiformis and the Agaricus bisporus is very well industrialized. And study mushroom development is not only can fulfill our curiosity, but also can benefit for the huge industry. So here, let's switch to the scientific part. So let's talk about the mushroom development from the mycelial stage. So under the proper environmental conditions, for example, the starvation, light induction, and temperature change, and so on, and it can trigger the mushroom formation, which can promote the mycelia aggregate with each other, and then we call this hypha knot, and then hypha knot can further development into the primordium. And from primordium stage, you can see the tissue differentiation. Basically, you can see the cap and the stipe, and basically you can find a preliminary shape of mushroom, and then primordium could be further developed into the mature mushroom. And in this topic, I would not dig into the morphological part, but I would like to introduce more from the molecular level. So at the very beginning, researchers just figured out the hydrophobin probably very important during mushroom formation. So what is hydrophobin? So hydrophobin is a kind of cell surface protein which localized at the hypha surface, which can help the mycelium here is the mycelium. It can help the mycelium to defend the humid substrate, to let the mycelium have a chance to escape from the humid environment and grows into the air. So once the mycelium grows into the air, and then different mycelium have a chance to meet with each other, and they can aggregate with each other and to form the hypha knot, and then further developed into the primordium. So this is a very basic knowledge we learn from the mushroom development. But based on this knowledge, we want to ask another question. So if the hydrophobins just covered at the surface of the mushroom, and then how mushroom can absorb the water from the environment? So taking this question, we did further analyze in our previous study. So previously we studied the Flammulina filiformis. So this is not common in Europe, but this mushroom is very popular in East Asia, and people like this mushroom. So we find the aquaporin gene was highly upregulated at the primordium stage and the young fruiting body type stage and the fruiting body type stage. So from this plot, you can find its clear expression pattern. It's just a peak expressed at the primordium stage and the young fruiting body type stage and the fruiting body type stage. So what is aquaporin? So actually people also call it as water channel protein. So it just localized at the cell surface, and the cell membrane is localized at the cell membrane, and it's just like a gate. Whenever it's open, it can let massive water entrance into the cell from the environment. So we also further studied this gene's expression pattern in other mushroom forming fungi. We find this gene also have the conserved development expression pattern during mushroom formation in other species. So this indicates us that the aquaporin dependent water absorption probably very important during the mushroom development. But the interesting part is aquaporin was not noticed by other researchers previously. So people just almost ignored this gene's function during mushroom development. They just focused on the aquaporin's function in the mycorrhiza fungi. So which means they focused on the plant root and the mycelium. They can form a symbiosis. They only focused on this gene's function in this symbiosis, which functions in water transportation. But in our previous study, we figured out this gene probably also very important during mushroom formation. So this is a very interesting very interesting in our previous study. So besides the water absorption, we find another interesting gene is the stipe elongation gene, ELN3. So we can say this gene's expression pattern have the similar expression pattern with the aquaporin gene. It's also peak expressed at the young fruiting body type and the fruiting body type stage. So what does this gene do? So let's have a look. People studied this gene in Coprinopsis scenario. So this is a wild type strain. You can see the stipe can innovate normally in the wild type strain. But if people mutate this gene in Coprinopsis, and you can see the mutant strain cannot innovate its stipe anymore. So this gene controls this stipe elongation during mushroom formation. So interesting part is we find this gene have the conserved expression pattern in other mushroom forming fungi. So another interesting part is another two mushroom forming fungi, they have this gene in their genome, but this gene just doesn't express, developmentally expressed. So this could be one of the reasons to explain why these two mushrooms doesn't have stipe, or let's say probably their stipe doesn't elongate. So this is interesting, and this also tells us probably this gene could be a potential target gene for further strain improvement based on molecular level. Because we know the Flammulina filiformis, people like the longer stipe, which means the large productivity. So we want to ask, so we are more or less sure there should have more genes which valuable for the further mushroom development, strain improvement for the mushroom development. And we also want to ask, there should have more genes which directly involved in mushroom development, but what are they? So probably we just start from the genome level is a good start point to find the important genes during mushroom formation. So based on our previous study, we find that different mushroom forming species have the conserved gene expression pattern during mushroom development. So those are different phylogenetically far related species, or genetically far related species, but they have the conserved gene expression pattern during the mushroom development, which includes the hydrophobins and lectins. So lectins, which functions the cell communication and the cell adhesion during mushroom development. So we based on genome data, we find an interesting part is based on this species tree of the fungal kingdom, we find, yes, the fungal kingdom, we find the important genes which related to mushroom formation, for example, the hydrophobins and the lectins and the white color complex and so on and so forth. They are really emerged, emerged earlier than mushroom emergency during the long evolutionary history. So which indicate us that there should have some higher rank regulatory factors to regulate those genes expression during mushroom formation. So we call this higher rank regulatory factor as transcription factor. So what is transcription factor? Transcription factor can bind the genes, those genes promoter region to regulate genes expression or regulate or just repress the genes expression. It's just like a commander. It can say, okay, at this time point, you start work and you stop and you do this and you do that. So under this well-organized regulatory pattern, and then it can regulate those building blocks. Let's say the hydrophobins and lectins and everything which necessary for mushroom development regulate properly to build the mushroom. So transcription factors is a really important gene in mushroom formation, we suppose. And if we look around, we just find the studies of transcription factors in mushroom development is really limited. And what about the target genes which regulate by the transcription factors also remains elusive. And this is not surprising in mushroom forming fungi, because we know the mushroom is very hard genetic manipulation. And previously, in one single piece of paper, only introduced the maximal 5 transcription factors. So this is compared with other model system, for example, like yeast, like fruit fly, and so on. This is quite a less number of genes which can manipulate in mushroom. So, but the lucky part is our colleague Matty has built the gene editing system in the model species Coprinopsis scenario, and which means we can easily to do gene knockout to interrogate the gene's function in mushroom, and nowadays more than 2,000 fungal genome released today, so which provide us a chance to high throughput interrogate transcription factors function in a large evolutionary time scale. So in our study, we selected 30 conserved transcription factors in mushroom forming fungi. We suppose they are very important, and we just want to knock out those genes in Coprinopsis scenario and to have a look, what about the mutant phenotype compared with the wild type strain, and try to figure out those transcription factors function in this process. And the interesting part is, among those 30 transcription factors, only 8 were functional studied. But for those 8 transcription factors, what about their target genes? And what about their functions mechanism during mushroom formation? We still do not know much about this. And another 20 genes have completely unknown function. So this is really interesting. We suppose they are very important because they are conserved in mushroom development, in mushroom forming fungi. So, but we just do not have an idea what does they do during mushroom development. So let's have a look about their expression pattern in Coprinopsis scenario. And we can say basically almost all of these transcription factors have upregulated at the early developmental stage. Let's say they upregulated at hypha-knot stage and primordium stage. So the hypha-knot stage and the primordium stage is a very important key transition point from the mycelium to mushroom. So once the hypha-knot and the primordium formed, and then the mushroom can be formed. So this is a very important stage. So we focused on the early developmental stage. We want to figure out what about those transcription factors function at early stage. So currently we knocked out about 25 transcription factors. This is one of the largest data set we can mutant number which we generated in mushroom forming fungi in our research. So I will not introduce them one by one in this presentation, but I will introduce some interesting phenomenon in our study currently. So firstly, I would like to introduce 2 morphogenesis transcription factors. They are Vol1 and BAC1. So they widely studied in Ascomycota species. So Vol1 was studied in the Candida albicans, is a animal pathogen. So Vol1 functions the cell type transition. So it functions from the white to opaque cell type transition, which means it's basically a morphogenesis transcription factor. It can regulate the cell shape change from the white to opaque. You can see the shape of the cell is different between these two. And another one is BAC1. Its function, this is a Neurospora, another model species in Ascomycota. This is Neurospora's fruiting body, and the BAC1 functions as the top structure of the fruiting body formation. We call it beak. And so these two are morphogenesis transcription factor. But what are the outlook gene of these 2 genes in mushroom forming fungi? Basically, people just didn't study it before, and we have no idea. So in our study, we knocked out of these 2 genes outlook in Coprinopsis, and we find the mutant strain, they cannot produce fruiting body anymore. So this is really interesting. We can find the gene which regulate morphogenesis in Ascomycota species, and they also have the important function in mushroom formation. And previously, people figured out those two genes, target gene, which includes the cell wall modification gene and hydrophin gene and the cell adhesion related gene, and in Candida albicans and in Neospora. And also from our previous study, we figured out that the cell wall modification and the cell-cell adhesion related genes was expensed in mushroom-forming fungi. So it's really interesting to investigate those two genes, target gene in mushroom-forming fungi. So now we do not know, but now we are working on that. So and and the second, I would like to introduce another two genes. So these two genes also upregulated at hypha-naught stage. And we suppose if we knocked out those two genes, probably also no hypha-naught and no mushroom formation. But to our surprise, if we knocked out those two genes, we find the mutant strand can produce more and dense primordia compared with the wild type strand. So this is really interesting. This indicates that these two transcription factors could be repressed during mushroom development. So we find these two genes homolog also upregulated at Flamina filiformis during its development. So in Flamina filiformis, people like dense primordium, which means these two genes could be the important target gene in further strand improvement, strand breeding based on molecular level. So this two gene is quite interesting and probably valuable. And the last but not least is let there be light. So I am not a believer, but I like this sentence a lot. This sentence comes from the Bible. And let's have a look how mushroom responds to light. So here is a dark condition. And once the light comes, you can see the two transcription factors, WC1 and WC2, just upregulated to response to the light. And these two genes was widely studied in Neospora. So let's have a look at this picture. So this is the wild type of Neospora. Once it sees the light, you can see the yellowish or orangish pigmentation can happen in the wild type strand. But if we put the wild type strand under the dark, you can see it's just nothing changed. It stay like white. So no pigmentation in wild type strand. So researchers just knocked out those two light response transcription factors. You can see the mutant strand, if the mutant strand put it under the light, basically it just keep the white color under the light condition. It cannot produce the orangish pigmentation, which means it just cannot see the light. So this is interesting. And what about these two genes? If we knocked out these two genes autolog in mushroom forming fungi, what will happen? So in our study, we knocked out those two genes in Coprinopsis scenario. So here is the wild type strand. If we put the wild type strand in the dark, so you can see only the elongation happens, but without cap expansion. So we call this as a dark phenotype. So if we put the wild type strand under the light, so you can see the elongation, stable elongation happens, and the cap expansion also happens, and the mushroom formed. But if we see the mutant strand, so the mutant strand, if we put them under the dark, they have the same phenotype with the wild type strand. Basically, they cannot, they do not see the light, and they produce the dark phenotype. If we put the mutant strand under the light, they still produce the same phenotype which they under the dark. So only elongation happens, but without cap expansion. So still remain the dark phenotype. So basically, they cannot see the light anymore. So this is very interesting. So, but what about the target gene of these two transcription factors? What gene of these two transcription factors regulate during the mushroom development is very interesting to study, but we do not have much knowledge about this. And in our study, we try to figure out this. And so from this picture, you can see those genes are functional studied previously during mushroom development. So basically what we know compared with each large data set, large genome data set, which contains a large number of the genes, what we know is really, really less. And we really do not know much genes which related to mushroom development at this time point. So compared with what we know, we really do not know much. But the good part is we are on the way. So people in our lab, we just study mushroom development from different aspects, and also have many lab all over the world. People just try to uncover the secret behind the mushroom development. Mushroom is tiny, it is not large, and it can grow very quickly, but to figure out the mechanisms behind is really, really hard. Probably 100 years later or 200 years later, but I believe finally we will figure out the mushroom development. And I would also acknowledge thanks all the colleagues in our lab with their help during my research. So I'm happy to take any questions. Basically, that's all of my topic. So thanks for your kind attention.
So, welcome online to the Mycology department. Thank you for that great presentation. I am personally amazed by the extent of your research and the state-of-the-art technology you used for it. Üdvözöllek a Mikológia Tanszéken, és hát köszönöm szépen ezt a nagyszerű előadást. Én személy szerint le vagyok nyűgözve a kutatásoktól, illetve hát azoktól a csúcsmodern technológiáktól, amelyeket ennek során használtatok. Here we are to answer the questions from our viewers, so we are looking for your questions, dear audience. Azért vagyok itt, hogy a kérdéseket tolmácsoljam, szóval várjuk a nézőink kérdéseit. First question, there was a figure in this in your presentation where I saw that more aquaporin is in the stipe than in the cap. Is it right? What is behind this, and is it general or specific to Flammulina? Szóval az egyik ábrán azt láttam, hogy több aquaporin van a tönkben, mint a kalapban, és hogy ez igaz-e, mi lehet-e mögött, illetve hogy ez specifikus, vagy pedig általános.
Yes. So actually, we say aquaporin gene upregulated at stipe is a phenomenon, and it's quite general or conserved pattern among the mushroom forming fungi.
Valóban az aquaporin a tönkben felülszabályozott, ezt általánosnak mondható, ez egy konzervált minta a gombák vonatkozásában.
So, well, about the mechanism, we don't know why it specifically expressed at a stipe tissue. But we can guess probably it's because stipe is a tissue of the mushroom which directly attach to the substrate. So we guess the stipe is a major part to absorb water in mushroom. So this is why this gene are specifically expressed in stipe. So this is a possible reason.
Nem tudjuk pontosan, hogy mi lehet ennek az oka, viszont hát egy tipp azért van ezzel kapcsolatban. Ugye a tönk az a szövettípus, amely még a szubsztrátumhoz kapcsolódik, és ezért fejeződik ki ott nagyobb mértékben az akvaporin. Thank you for your answer.
Thanks.
You mentioned mentioned about strain improvement at molecular level, and what do you think whether the consumer are ready for a genetically modified mushroom product, a food product? Szóval említetted ezt a bizonyos fejlesztést, amit molekuláris szinten lehet akár végrehajtani, és hát vajon, mit gondolsz, vajon a vásárlók felkészültek-e már, illetve fogadnák-e a genetikailag módosított gombaterméket, élelmiszert?
So I think global-wise, different part, maybe people have different opinion. Europe much more strict, but in China and in US, I think relatively acceptable. Yeah.
Hát globálisan nézve a különbségek fedezhetők fel. Európa ebből a szempontból mereven áll hozzá a kérdéshez. Kína és az Egyesült Államok elfogadók ezekkel a technológiákkal kapcsolatosan. Okay, thank you. You mentioned 100 years to fully reveal the secrets of mushroom forming. Is it your optimistic or pessimistic estimate?
Thanks for this question. So I think finally I'm optimistic about we can reach the end, reach the final answer, but it have a long way to go. Because, for example, about 100 years ago, Darwin just comes out an idea about evolution. 100 years later, people still on the way, still study evolution. So finally we will reach there, but it will have a long time.
A kérdés úgy hangzott, hogy említettél, hogy 100 év szükséges esetleg ahhoz, hogy hát teljes mértékben felfedjék a kutatók, felfedjétek a gomba termőtestképzésnek a titkát, vagy titkait, és hogy ez vajon egy optimista, vagy egy pessimista becslés, hangzott a kérdés, és a válasz pedig optimista becslés, abban az értelemben, hogy végül elérjük a célunkat. Az út az hosszú lesz, és nyilvánvalóan az evolúció kutatása az folyamatosan zajlik, tehát bizonyára még 100 év múlva is kutatjuk ezeket a kérdéseket. So, that was my last question. Thank you again for your brilliant lecture. And on the long road of your research, when there are partial results, please come back and show it in the Mycology department.
Okay.
Szóval ezen a hosszú úton, ami a kutatásodat jelenti, hogy ha vannak részeredmények, akkor légy szíves, hogy gyere vissza, és itt a Mikológia Tanszéken mutasd ezt be számunkra. Thank you for the kind attention for our audience. We will meet the second Wednesday of the next month. Thank you and goodbye.
Thank you.